This screencast discusses the neuron, which may be found in Chapter 7 of your textbook. This screencast was designed to help you achieve the following objectives. Describe in detail the structure of a neuron and the functions of its various parts. Define nuclei and tracts. Define ganglia and nerves. Describe white and gray matter. Explain the functional classifications of neurons. And lastly, explain the structural classification of neurons. Now that you have a good understanding of neuroglial cells, now let's turn to discuss the second type of cell that is found in nervous tissue, the neuron. Neurons are commonly referred to as nerve cells. Neurons are specialized cells. They're specialized to transmit messages and therefore communicate with other cells of the body. Neurons do not have centrioles and therefore cannot divide. Also, nervous tissue do not contain many stem cells that have the ability to regenerate new neurons. So for the most part, when neurons are destroyed, they're lost forever and are not replaced. Let's now look at the anatomy of the neuron. The neuron can be divided into two major regions. The two regions of the neuron include the perikaryon, also known as the cell body or the soma. Different books use different terms. It does not matter to me which term you use. It is simply the cell body of the neuron. This is where the nucleus is found, and all of the organelles that you have become familiar with, if they're in the neuron, they're also found in the perikaryon. The perikaryon is the metabolic center of the cell. Extending from the perikaryon are processes. Processes are fibers, and they extend from the cell body. First, let's talk in more detail about the perikaryon. One structure found in the perikaryon is the Nissl substance. Now, the Nissl substance is basically a specialized, compartmentalized, rough endoplasmic reticulum. Another structure I'd like to call your attention to are the neurofibrils. The neurofibrils are the filaments of the cytoskeleton, and they help determine and also maintain the shape of the neuron. In lab, you looked at nervous tissue, and one of the things that you should have noted was that neurons have a very large nucleus with a large nucleolus. Now that we have discussed the cell body, let's now turn our attention to the processes. Again, processes are nerve fibers that extend from the cell body. Dendrites are processes that detect stimuli or changes, and in response to the stimuli, conduct electrical signals toward the cell body. Axons are much longer processes that conduct electrical signals away from the cell body. Although neurons can have many dendrites, they typically only have one axon extending from the cell body, although collateral branches can branch off of that one axon. If we look more closely at the axon, you'll notice that the axon begins at a funnel-shaped structure called the axon hillock, and it ends as axon terminals. These are enlarged endings to the axon. These axon terminals contain neurotransmitters, and those neurotransmitters are going to play very important roles in allowing the neuron to communicate with other cells. If you recall, oligodendrocytes and Schwann cells form coverings that surround the axon called myelin sheaths. In this particular example, the myelin sheath is formed by Schwann cells. Notice that there are little gaps between adjacent Schwann cells where the axon is exposed. These are called nodes of Revere. If we were looking at an axon where the myelin sheath was formed by oligodendrocytes, you would also see nodes of Revere. We will talk later on the function of nodes of Revere when we discuss how nerve impulses travel down the axon. To highlight the importance of the myelin sheath, multiple sclerosis is an autoimmune disease where one's immune system attacks a particular protein in the myelin sheath, effectively destroying the myelin sheath. And as a result, the individual increasingly loses his or her ability to control their muscles. Have you heard the terms gray matter and white matter with respect to nervous tissue? White matter consists of myelinated nerve fibers. Notice that the myelin sheath is white in appearance, at least in comparison to the cell body. So white matter basically consists of dense collections of myelinated nerve fibers or myelinated axons. You find gray matter where you have lots of cell bodies. Again, because the cell bodies are gray in comparison to the myelinated axons. You will also find some areas of the body where there are axons, but they're unmyelinated, and they also would make up the gray matter. So for the most part, the difference between white matter and gray matter is simply the presence or absence of myelinated axons. Neurons do not exist singularly. They are arranged in clusters. In the central nervous system, clusters of cell bodies are called nuclei. So much of the gray matter in the central nervous system are nuclei. The axons extending from the nuclei are bundled together in collections called tracts in the central nervous system. Most axons in the central nervous system are myelinated, therefore these tracts would appear as white matter. We have the same situation in the peripheral nervous system, but we use different terminology. Clusters of cell bodies in the peripheral nervous system are referred to as ganglia, and they also would appear as gray matter. And the axons that extend from the ganglia are bundled together in collections called nerves. And again, most, if not all, of the nerve fibers, the axons in the peripheral nervous system are myelinated, so nerves would also appear as white matter. Let us now talk about how we classify neurons. Neurons can be classified functionally by the direction in which they send electrical signals with respect to the central nervous system, or they can be classified structurally by the number of processes that they contain. To illustrate the functional classification of neurons, I'm going to use the following figure. What you're looking at here is a cross-section of the spinal cord. The spinal cord, of course, is a part of the central nervous system. Neurons are classified functionally by the direction in which nerve impulses are sent in relation to the central nervous system. So I'm going to illustrate the functional classification of neurons based on the direction in which nerve impulses are sent with respect to the spinal cord in this figure. Sensory or afferent neurons send electrical signals from receptors in different parts of the body to the central nervous system. In this figure, the sensory neuron is shown in blue, and this arrow shows you the cell body of the sensory neuron. So we have dendrites or receptors in different parts of the body that are responsible for detecting stimuli. The dendrites could be in the skin, they could be in muscles, they could be many places of the body. In response to a stimulus, an electrical signal is sent toward the central nervous system. Again, sensory or afferent neurons send electrical signals from receptors in different parts of the body to the central nervous system. Motor or efferent neurons carry electrical signals from the central nervous system out to effectors. In this figure, the cell body of the motor neuron is located in the spinal cord, and it will send electrical signals down its axon out to muscles and glands in different parts of the body. So again, motor or efferent neurons carry electrical signals away from the central nervous system out to effectors. The third and final class of neuron are the interneurons or association neurons. These neurons are found in the central nervous system, and they send electrical signals within the central nervous system. Effectively, they connect the sensory neuron to the motor neuron. This is where integration occurs in the central nervous system. Neurons are also classified structurally by the number of processes that they contain. Multipolar neurons contain multiple extensions from the cell body. If you look at this figure, you can see that while there's only one axon, there are many dendrites. So this is a multipolar neuron. Notice that all motor and interneurons are multipolar neurons. Going back to this figure, notice that both interneurons and motor neurons have multiple processes, many dendrites in this particular example. Therefore, they are multipolar neurons. The second structural class of neurons are the bipolar neurons. Bipolar neurons have exactly one axon and one dendrite, so two processes. Bi means two. Bipolar neurons are rare in the body, only being found in some special sense organs such as the eyes. The third and final structural classification of neurons are unipolar neurons. Unipolar neurons have one single process shown here. Uni means one. That process does branch into a peripheral process which extends away from the central nervous system out into the body, and a second central process that extends toward the central nervous system. But note, they are all branches of that short single process. One mistake the students make in lab is to call this a bipolar neuron. It is not. It is a unipolar neuron where you have that one short process where the arrow is pointing, and then it branches into two branches. If we go back to this slide again, notice that the sensory neuron is a unipolar neuron. It has that one short process that then branches into a central process extending toward the central nervous system and a peripheral process extending toward different parts of the body. So all sensory neurons are unipolar neurons. Now let's review the objectives that this screencast was designed to help you achieve. Describe in detail the structure of a neuron and the functions of its various parts. Define nuclei and tracts. Define ganglia and nerves. Describe white and gray matter. Explain the functional classifications of neurons. And explain the structural classifications of neurons. The next screencast discusses the resting membrane potential.